This video is going to go over nucleic acids and mainly focus on their structure, and then we'll look at transcription, translation, and DNA replication in a subsequent video. So both DNA and RNA are what we call polynucleotides. It's one of the biopolymers that we mentioned when we introduced biochemistry, and they're formed through condensation reactions just like all the other biopolymers. The monomer of polynucleotides is called a nucleotide. But what's a little bit trickier about this one is that the nucleotide itself is actually composed of 3 different distinct parts, which have already condensed together to form the nucleotide in the first place. So the first thing we're going to cover is what makes up a nucleotide. So every nucleotide has 3 parts. Part 1, a 5-membered sugar that is in a ring form. It could also be called a pentose sugar. And the 2 different sugars that you need to know about, and it's what gives the D or the R in both DNA and RNA, is deoxyribose and ribose. You can see the difference is simply that at this carbon here, in one of them there's an OH, and in one of them there's just an H, hence the deoxy, meaning it's missing one oxygen. So they're both sort of like ribose, but one of them is missing an oxygen. You start the numbering of any ringed sugar at the carbon after or to the right of the oxygen that's in the ring. So that's number 1, 2, 3, 4, and 5 up here. And the same goes for this ring. Okay, that's going to be very important that you know those numbers, because especially the number 1, the number 3, and the number 5 are going to be very important spots when we form up our nucleotides. So this is the first part. These are in the data booklet, and it should be pretty easy to remember which one is in DNA and which one is in RNA. Okay, the second component is a phosphate group. It's actually originally phosphoric acid, so that's H3PO4, but when it's bound up into DNA, one of the hydrogens here is gone, and this is bound to one of the sugars on one side, and then this hydrogen is gone through a condensation reaction to a sugar on the other side, and then this remaining H is actually, because the environment of most cells is going to be pretty close to neutral, this H is actually dissociated off as an H+. And so even though it comes from phosphoric acid, the phosphate group, when it's bound up to DNA, what you actually end up seeing really is a bond from that oxygen to a sugar, a bond from that oxygen to a sugar, and this oxygen with a negative charge remaining. So the reason why DNA and RNA are called nucleic acid is actually because of this part, this phosphoric acid group, and this donation of a proton. It's a little confusing though, because the way that we generally look at the structure, that proton has already been donated. But it is where the name nucleic acid comes from. It's from this phosphoric acid group. You should call it a phosphate group, and it will have 1 net negative charge. The third component of the nucleotide is the nitrogenous base, the famous 4-letter code A, G, C, and T, or U if we're dealing with RNA. The nitrogenous bases can be split into 2 different categories, the purines and the pyrimidines. Now, you are expected to know which is which. The way I remember is with some very silly little things I've come up with, which is when I hear purines, I think of the word pure, and things that I think of as pure would be like silver and gold, these elements. So pure silver, Ag, reminds me that adenine and guanine are the purines. Pyrimidines, another very silly, dumb way to try to remember them is that pyrimidines makes me think of the pyramids, which makes me think of big giant stones that had to be cut. So C, U, and T, cytosine, uracil, and thymine are the pyrimidines. I don't know what you're going to use to try to remember it, but that's my way. Those are my helpful tips. If you can remember just one of them, then you can figure out the remaining ones if they're not in that first category. So if you can just remember purines is Ag, then the other ones must be pyrimidines. Okay, so comparing DNA and RNA. So they do have a number of similarities, but they also have a number of differences. We've already gone over that they have a different sugar, deoxyribose, versus ribose. Those nitrogenous bases, 3 of them are always the same, but in DNA, there's thymine, and whenever there would have been a thymine, when you're in RNA, there's uracil, which is a slightly different, it's very similar in structure to thymine, but slightly different. So uracil is the nitrogenous base that replaces thymine when you're in RNA. DNA is a double helix structure, so it has 2 strands that are held together, whereas RNA is always single-stranded. And DNA only stays in the nucleus, whereas RNA can travel from the nucleus to the cytoplasm and the ribosomes. So the first 3 are really structural differences, whereas these ones are more just properties that are different. Okay. If we're going to try to build these nucleotides and put them together, I'll save this for class, but we'll go on to the next page and we'll kind of review it. But you're going to try to put together some sample nucleotides in class between nitrogenous bases, sugar rings, and the phosphate groups. All of these structures that are on the page here are given in the data booklet, so it's just a matter of sort of placing them in the right place and doing some condensation reactions, but we'll give that a try in class. All right. The forming of the backbone. So you hear of the double helix. That means there's actually 2 strands of DNA that are then attracted together. So those 2 strands themselves are actually quite strongly held together, and they are bonded up by condensation reactions. And so the backbones of both DNA and RNA are held together by covalent bonds. So in terms of biochemistry, we're going to consider that to be fairly permanent in terms of how they are held together. The condensation reactions that hold the backbone together occur at carbons 3 and 5. So if this is 1, that's 2, this is the 3rd carbon, so that's one of the carbons we're going to be looking at, and 4, 5, this is another one of the carbons that we're going to be looking at. So what happens is this OH undergoes a condensation reaction with that phosphoric acid, and so on this side, this carbon is going to be bound up to an oxygen that's bound up to that phosphorus with the O on one side with the negative charge, the O here that doesn't have a negative charge, the double bonded O, and then this one which leads to the next sugar. So there's the phosphate group. So there's one condensation reaction. Remember that this was phosphoric acid, so it began with an H, hence here's the water that's going to get removed when that links up. And then right here, same thing, but on the other side, you're going to have the phosphorus again, which originally has the O and H, and the OH from the sugar and the H from the phosphoric acid group are going to be removed. And so you're going to have this bond straight from the carbon to that oxygen, and then a repeat structure of the phosphate group right there. So it's pretty straightforward. By now, you should be very familiar with condensation reactions, removing water, linking 2 molecules together. The key to remember is where they happen. So it's off of the 3rd carbon and off of the 5th carbon, and it's phosphate on either side. So each sugar is bonded to 2 different phosphate groups, and each phosphate gets bonded to 2 different sugar groups. The nitrogenous base is not part of the backbone. The nitrogenous base is going to stick off of this first carbon. So we'll just say to the NB, the N base. The nitrogenous base will go off the number 1, but that's not what we're looking at right now. The backbone itself is just phosphate, sugar, phosphate, sugar. The bond here is called a phosphodiester bond. It's a pretty cool name. And if you think about it, you've got a phosphorus group, right? And the phosphorus group is forming the bond here to this carbon, and it's forming the bond here to that carbon, and it almost looks like an ester. If this P was replaced with a carbon, then you would have like an ester functional group right here. You'd have COOC, kook, your kooky old Aunt Esther. But because it's a phosphorus, then it can't just be called an ester linkage. So it's called a phospho for phosphorus, and it's like 2 esters, one on either side of the phosphorus. So it's called a phosphodiester linkage or a phosphodiester bond. That's the special name for that. We've talked about, there's peptide bonds, there's glycosidic links, and now the one to know in DNA is the phosphodiester bond. It's a very strong covalent bond, so the strands are really not going to be broken down, especially not during normal DNA or RNA activity. and we already said that the bases are not part of the backbone. Sometimes you hear DNA referred to as 5' or 3', having different ends, and that simply refers to that number carbon. So if you are at the end where you end near the number 5 carbon, then that's the 5' end of DNA. That's where that name comes from. And if you're at the 3' end, then you're near the number 3 carbon end of the chain, whether that's a phosphate group here or it's just the last sugar. And so that numbering system can be used for RNA or for DNA. They both have the sugars numbered in the same way. So either at the 3rd carbon is at the end of the chain, or the phosphate off the 5th carbon is at the end of the chain. That's where 3' and 5' numbering system comes into play. Okay, last structural thing that we're going to talk about. No, I guess there's 2 more structural things we're going to talk about in this video. Okay, so the reason why DNA and RNA work so well as a storage unit for genetic information is that the nitrogenous bases are highly specific for what they will attract. That's how the system is going to work. So the 2 strands in DNA are held together only by hydrogen bonding between the nitrogenous bases. But each nitrogenous base can only hydrogen bond with one other nitrogenous base. So they have a complementary pair that they form. They have base pairs, or they have like sort of a partner that they can hydrogen bond with. So it turns out that there's always going to be a purine bonding with a pyrimidine. That's how they end up fitting together. Guanine and cysteine always hydrogen bond together, no matter if you're talking about DNA or RNA. And they are able to form 3 hydrogen bonds. So I remember that G and C both rhyme with 3. Okay, so G and C are going to always be able to form 3 hydrogen bonds. And the IB has actually been pretty nice in how they draw the structures of the nitrogenous bases. What you're going to look for is at the top left of every structure, there are 3 things that can form hydrogen bonds. So here's a H enough, N to H, and here's an N that has a lone pair, and here's an O that has 2 lone pairs. So both of these 2 things need a hydrogen from the other molecule. This one does not need a hydrogen from the other molecule. If you look over here at the top left, notice this N has that H enough that we needed. This one has the H enough that we needed, and this one has a lone pair because we didn't need an H. So in the end, this H is going to be able to form a hydrogen bond here. This N will be able to form a hydrogen bond with that H. This O, a hydrogen bond with one of those H's. So over here, I did a little inversion. I flipped the image, and you can see that they sort of perfectly line up here. So there'd be a hydrogen bond between the O and the NH, between this H and that N, and this H and that O. So C and G can form 3, and there's only these 3 spaces that line up, the top left in their structures in the data booklet. Okay, then there's, in DNA, A always binds up with T through hydrogen bonding, and A always binds up with U in RNA. So the T and the U trade places. And I remember that A, T, U, well, if you read that, you'd say a 2. They form 2 hydrogen bonds, a 2. So again, you're looking at the top left. So top left and top left. And notice that once again, they're sort of like perfectly matched. This one has an H and this N doesn't. This N has an H, this O doesn't. Those are where the hydrogen bonds are going to form. So that's drawn down here. Here's the H to that O, and here's this N to that H. Those are the H bonds, which should be drawn as dashes should you try to draw these things on the exam. It's going to work the exact same way in uracil. You can see that uracil and thymine really are basically identical molecules, except that for this methyl group, where this one has an H. So it's going to be, the hydrogen bonding really doesn't change still between those, and I think at this point you get the idea. But you should know the number of H bonds that occur between the different base pairs, which bases go together, and if requested, you should be able to draw in the hydrogen bonding between the bases given the structures. Okay, now we're at the last structural thing about DNA and its properties. What one thing that's really cool about DNA is the way that it is in this double helix, the very famous double helix. That's actually super useful for protecting the genetic information. Um, the If you look at the backbone is quite polar. It's made of sugars which have OHs, and it's made of phosphate groups that have a negative charge. So the outer backbones are rather hydrophilic. They are polar, and therefore they're water soluble. So the outsides of the molecule are quite water soluble. The inside, these nitrogenous bases are a lot more nonpolar. C to N bonds are fairly nonpolar. There's a lot of just carbon in the rings. So mostly on the inside, they are hydrophobic. And so naturally, when they're in the aqueous environment of the cell, they form this coil, this double helix. And it's really important because all of the genetic information is stored in the nitrogenous bases in the exact order of all those A, T's, C's, and G's. And so by coiling up and keeping those at the center, it actually keeps the sequence of bases relatively protected from whatever molecules may be knocking into the DNA when it's inside of your cell. So it's a really cool structural feature of DNA that the way it coils up actually makes it very stable and very good at protecting that perfect genetic information that's all lined up. The other cool thing about DNA is that because it has a negative charge, because of those phosphate groups, it actually ends up being highly attracted to proteins that are positively charged within the cell. Those proteins are called histones, and so the negative and positive charge is just a simple attraction that's going to take place, and so the DNA ends up wrapping around these histones within the cell, and that's how the DNA structure gets stabilized and wrapped up into the form that we know of as chromosomes, which is how they coil up before cell replication. So just a couple other really neat things about DNA structure, why it works so well. It's got this overall negative charge. It has the polar things on the outside, and it has the hydrophobic things on the inside. So it coils to protect its genetic information. It wraps with positively charged histones to give it that chromosome structure. And the last bit, which I guess I skipped over, is that when the DNA strands go together, they are called anti-parallel, meaning that that thing we talked about, 3 and 5 prime, those are always going in opposite directions. So one strand's going 3 to 5, while the other strand's going 5 to 3. And that's all the structural information that you need to know about DNA and RNA. You should be able to compare the two, contrast the two, and be able to explain and draw how the sugar phosphate backbone is formed and how the nitrogenous bases hydrogen bond together. One thing that I do believe I skipped over is when you are drawing in the bases, if you're going to attach them to the chain, the one that forms the condensation reaction, with the sugar, is this very bottom nitrogen. So the one that's going to form the condensation reaction, so you've got the sugar here, it's this OH group that's going to condense with the nitrogenous base. So you need one more H to form that water molecule. And so the H's that are going to condense are these H's at the bottom here. So those are the hydrogens that would leave, and then the bond to the sugar ring would be off of that bottom nitrogen. And so that's going to be the same on all of these. I apologize, I didn't do that before. But in terms of actually linking the base up covalently to the rest of the backbone, it's always that bottom right nitrogen. It's going to lose that H, and that's where it's going to covalently bond up to the sugar ring.